Okay, so this is going to be chapter 10, and I've decided to make this particular chapter extra credit. So we're going to have exam 5. That's going to be like a regular exam, only worth 50 points, and it's completely voluntary, whether you want to take it or not. It's going to be on this information here on how DNA works and genetic code and a little bit about viruses. So here we go. Let's talk about chapter 10 and the structure and function of DNA, how DNA actually works, which comes down to protein synthesis, or actually also gene expression. Really, protein synthesis and gene expression are really the same thing, because when you express a gene, you make the protein that it codes for. And we're going to look at the genetic code and how genes actually code for proteins in this chapter. All life on Earth shares a universal genetic code, so your DNA could be used to genetically modify bacteria and vice versa. This is how we make transgenic organisms, organisms that have genes from other species in them that actually work, because all life on Earth uses the same genetic code. So all genes and all organisms work the same way. It's part of the unity of living things. Okay, it's global threat of Zika virus. So we have these, what do you know? Emerging viruses like coronavirus. 2015 we had Zika virus. And these are new viruses that come out. So in order to understand how viruses work, and viruses are really simple things. They're crystals. They're not living things, but they hijacked our cells to do their bidding, which is to make new viral particles. And so you understand how viruses work, you need to know how genes work. And so we're not going to go into Zika viruses too much, but remember, like all viruses, it's relatively simple. It's an infectious crystal constructed of a molecule of nucleic acid. Well, the simplest viruses are a molecule of nucleic acid, and all viruses have a molecule of nucleic acid surrounded by a protein coat. Sometimes there's a little bit of phospholipid on the outside, so it almost looks like a cell, but it's not a cell. There's an illustration of Zika virus. Okay, so DNA was known to be a chemical component of cells by the late 1800s, but Mendel and other geneticists had no idea what role it played. They didn't know that DNA was the genetic material. They actually thought that maybe protein was, and there was some elegant experiment to show that yes, DNA is the genetic material. But they didn't know how DNA looked like and how it worked. So what gave DNA its unique properties, the capacity to store genetic information, copy it, and pass it on from generation to generation. So that information is passed on from generation to generation. So as DNA is passed on, it's the genetic material for all life on Earth. Some DNA, some viruses use RNA as their genetic material, but they're not living things. So it's appropriate to say that for all living things, DNA is the genetic material. Okay. The nucleic acids, long chains of polymers of nucleotides. So they could be called polynucleotides. And polynucleotides can be very long and have any sequence of the 4 different types of nucleotides. Well, a different type of nucleotide. A nucleotide consists of a sugar, either ribose or deoxyribose, a phosphate group. And that's true for, all DNA has the same sugar, same phosphate group. On the other side of the sugar is a nitrogenous base. And there's 4 different nitrogenous bases in DNA. They all begin with a different letter, so they're just designated with the first letter: adenine, cytosine, thymine, and guanine. And so they're joined together by covalent bonds between the sugar of one nucleotide and the phosphate of the next, in a repeating pattern of sugar, phosphate, sugar, phosphate. So it's called the sugar phosphate backbone. And that is the same for all DNA molecules as they have exactly the same sugar phosphate backbone. That's not what makes one DNA different from the next. It's the order of the nitrogenous bases, the orders of the A's, the T's, the C's, and the G's. That's where the information is. DNA is an informational macromolecule, and that's where the information is located in the sequence of those nitrogenous bases. So nitrogenous bases are arranged like ribs that project in its backbone. Each nucleotide consists of, so as I mentioned, each nucleotide consists of a nitrogenous base, a sugar, and a phosphate group. Okay, the sugar in DNA is called deoxyribose. As opposed to ribose, it's missing an oxygen atom, so it's a slightly different sugar. So DNA is deoxyribonucleic acid. Okay, the chemical structure of a DNA polynucleotide. There's DNA double helix. Yeah, DNA is always, in living things, it's always double-stranded. In some viruses, it can be single-stranded, but in living things, it's double-stranded. And it can be drawn with this blue and yellow sugar phosphate backbone, and here are the nucleotides projecting out. And so here is a nucleotide. There's a sugar, there's the phosphate, there's the nitrogenous base. So the four DNA nucleotides differ in their nitrogenous bases and combined into two groups. Thymine and cytosine are single ring structures. Adenine and guanine are larger double ring structures. Now, instead of, now we're going to look at some differences between RNA and DNA. One difference is the R versus the D. RNA has ribose for sugar, and it can be double-stranded or single-stranded. Also, instead of thymine, RNA has uracil in place of U, in place of thymine. So there's no thymine in RNA, there's uracil instead. So there's an RNA polynucleotide. Here it is single-stranded, and lots of DNA. We're going to look at, and there's one type of DNA, you know, it can have different sequences, but we're going to look into at least 4 different kinds of RNA, and it's actually more than that. But we're going to look at least 4 different types of RNA later on. So compare and contrast the chemical components of DNA and RNA. DNA has deoxyribonucleic acid, RNA has RNA has deoxyribose for sugar, and RNA has ribose. DNA has thymine, no uracil. RNA has no thymine, but has uracil. And DNA is always double-stranded. RNA can be double-stranded or single-stranded, depending upon the type of RNA we're talking about. So we're not going to go into too much about the history of how Watson Crick, a very famous scientist who back in the 1950s figured out that DNA is a double helix. And they did so by looking at X-ray crystallography photographs made by a famous woman, Rosalind Franklin, who took the photographs. And back then and now, all the best X-ray crystallographers are women. Nobody knows why. And she was the best of the best. There was a group in Caltech, led by Linus Pauling, who was trying also to figure out the structure of DNA at the same time, because they knew the lab that figured it out would get a Nobel Prize. He didn't have quite as good an X-ray crystallographer as Watson Crick did in Rosalind Franklin. Of course, Rosalind Franklin hated James Watson. He was an American, Francis Crick was a Brit, and he's kind of a jerk. He's real arrogant, and she didn't care for him, so she wasn't going to show him her pictures. But the pictures were made in the lab of Maurice Wilkins. He was the head guy in the lab. So he went over and took her pictures and gave them to Watson and Crick. And that's how they figured out using wire models that it was a double helix, the double helical molecule with the nitrogen bases pointing to the inside. Linus Pauling got it wrong. He thought it was a triple helix with the nitrogen bases pointing outside. He was wrong. Watson and Crick got it right. And there they are, Watson, Crick on the left and Watson and Franklin on the right. So DNA is like a full ladder twisted into a spiral. The sides represent the sugar phosphate backbone. Each rung in a ladder is a pair of bases connected by hydrogen bonds. And as base pairing rules dictate the side-by-side combination of bases that form the rungs in the double helix. We'll look at the base pairing rules. Here's the rope ladder model of the double helix. Right? So we're going to look at the detailed representations of double helix in these figures coming up here. There's the ribbon model here, the atomic model, the computer model. The atomic model, you can see, well, here, ribbon model and atomic model, you can tell the base pairing rules. A binds with T with 2 hydrogen bonds. G binds with C with 3 hydrogen bonds. Those are called the base pairing rules, or also Chargaff's rules, because there's a guy, Dr. Chargaff, actually figured it out. Okay, so 1953, they published a paper proposing a molecular model for DNA and suggesting how DNA could be replicated. Was that 9 years later? Usually it takes 20 years for work to be done and recognize how important it was and get a Nobel Prize for it. Only 9 years. Watson, Crick, and Wilkins received the Nobel Prize for the work. Rosalind Franklin died of cancer in 1958, and therefore tragically was not awarded the Nobel Prize. She would have been had she lived, because in contrast to other awards like Academy Awards, who they give posthumous Academy Awards, they don't give posthumous Nobel Prizes, so she didn't get it. Okay, so in the paper, they proposed immediately that the structure of DNA, immediately proposed a possible copying mechanism for the genetic material. So the structure of DNA points toward a molecular explanation for life's unique properties of reproduction and inheritance, and arrangement of parts within DNA affects its actions within the cell. An example of relationship to structure to function. So remember, when a cell reproduces, it has to duplicate its DNA, right? Providing one copy to each to the new offspring cells. So Watson-Crick's model suggests that each DNA strand serves as a template for a new strand. And this is called a semi-conservative means of DNA replication. So you start off with one strand, it breaks apart, and then use a template of the 2 strands to make a new strand. So each new strand of DNA is a combination of an old strand and a brand new, newly constructed strand. Okay. So we know the sequence of bases on one strand of DNA. You know the sequence of bases on the other strand, on the complementary strand. Because you have an A here, you put a T here. You got a G here, you put a C here. You got a T here, you put an A here. You have a G here, you put a C here. A's and T's, C's and G's. So there's the DNA replication. The DNA parent molecule is by enzymes is taken apart. The hydrogen bonds are broken, and then a molecule called DNA polymerase takes nucleotides that are pre-made and takes them and places them in the proper places in the old strand that's now opened up. So like I said, if it's an A here, the DNA polymerase puts a T here. If there's a G here, the DNA polymerase puts a C here, and so on and so forth. And you make these two new daughter DNA molecules where one strand is old, the blue, and one strand is brand new, made by the DNA polymerase, which is in orange. So as I said, DNA polymerases are the enzymes that make new DNA molecules. So DNA replication becomes at a double helix at specific sites called origins replications, and it proceeds in both directions. So DNA replication ensures that all the body cells and multiple organisms carry the same genetic information. And also a means by which genetic information is passed along to offspring. Now we're going to see that DNA polymerase is a very accurate molecule. It's accurate. It copies DNA almost perfectly, but not perfectly, because remember, nothing is perfect. So it makes mistakes, and those mistakes are very important. They're called mutations. And we're going to look at later on the mistakes kinds of mistakes that are made and the consequences of those mistakes. And then we get into evolution, because mutations are the working material for evolution. Okay, here's these multiple bubbles and replicating DNA. We have a origin replication, and you make two new strands. Checkpoint: how does complementary base pairing make DNA replication possible? Well, because the DNA polymerase knows if there's an A here, you put a T. It knows the base pairing rules, and it just follows them. Okay, so DNA provides instructions to a cell and an organism as a whole. So here's where you link genotype to phenotype. Origins genotype, its genetic makeup. What alleles does an organism have at all its gene loci, right? That's the heritable information containing a sequence of nucleotide bases in DNA. The phenotype of the organism's physical traits arises from the actions of a wide variety of proteins. So the proteins made from the information in its gene determines an organism's phenotype, how you look on the outside, what kind of how these genes are expressed, what genes are expressed and how they are expressed is the phenotype. Okay, DNA specifies the synthesis of proteins in two processes: transcription. What's transcription? Just a copy. The transfer of genetic information from DNA into an RNA molecule, right? And then translation, the transfer of information from RNA molecule into a polypeptide, a protein. And it's called translation. It's because there's really two languages we're looking at here: the language of DNA and the language of protein. They're different languages. And so in order to make a protein from the language of DNA, nucleic acid, RNA, you have to translate it. So you have to think of, I love this analogy as if the DNA are books in the library, all right? And the genes are the pages in the books in the library. But this is not a lending library. You can't take the books out of the library. All you can do is go into the library, open one of the books, and copy one of the pages. Then you close the book and you put it back on the shelf. And then you bring the copy out of the nucleus and bring it to a ribosome to be translated, because the books are in a different language. They have to be translated, and they're translated at the ribosomes. So this information is translated at the ribosome to make the protein that's coded for by the gene that's in the book in the library. Okay, the function of a DNA gene is to dictate the production of a polypeptide. The relationship between genes and proteins is thus one of information flow. How does information flow? Flows from genes in the nucleus out to ribosomes, out to an actual protein that you made. So remember, a gene is a recipe for a protein. That's in a different language. So it's the flow of information, of genetic information. transcription from DNA to RNA, then the RNA leaves the nucleus, goes to the cytoplasm, goes to a ribosome, and is translated there. That information is converted into a specific protein that has a specific order of amino acids in it that came from the information in a gene in DNA. Okay. So what's a language of nucleic acids? All right. What do you need for a language? Well, let's go. What do you need for a Western language? I'm not talking about the Eastern languages that don't have an alphabet. Western languages have alphabets, and you need an alphabet. So what is the alphabet of DNA? It's A, T, C, and G. And then what do you need? You need words in sentences. All right, the words in DNA are always only 3 letters long. Right? And then you need punctuation. We'll look at where do you begin the message, where do you end the message. So you have a sentence with words, and you have a capital letter and a period to tell you where the message starts and the message ends. And we're going to see what those are. So the language DNA is written as a linear sequence of nucleotide bases, A's, T's, C's, and G's. The typical gene is a few thousand nucleotides long. Because we're going to see that in order to, that each word in DNA is 3 letters long. So if you're going to have, and that codes for 1 amino acid. So if you're going to have, if you want to code for a protein that's 100 amino acids long, you're going to need 300 nucleotides to code for. Okay. When a segment of DNA is transcribed, the result is an RNA molecule. Process called transcription because you've just copied. Well, it's a complementary copy, right? Of one strand of DNA. Complementary means it's the other nucleotide. So on the strand, on a coding strand is a T, on the complementary strand was an A. You know, on the coding strand is a G, the complementary strand is a C. Okay. And in copying RNA, DNA, if you see a on the coding strand, it's not a T for the RNA, it's a U. In RNA, remember, A binds with U, not with T, because there's no T in RNA. Okay. The sequence of nucleotides of the RNA molecule dictates the sequence of amino acids of a polypeptide. All right. So, and experiments have verified that the flow of information from gene to protein is based on a triplet code. Because here's the thing. So you want to designate an amino acid in a DNA molecule, where it can't be a one-to-one correspondence because remember there are 20 amino acids that have to be coded for. There's 20 amino acids that can be that are found in 20 different amino acids found in proteins. So you have to code for those. So you can't have a one-to-one code because there's only 4 different letters in DNA that only code for four amino acids. What about a 2 nucleotide? How about 2 bases, like A or T or C or G or GG? That's not enough either. There's only 16 of those, and you need 20. So it's a triplet code. 3 letters in DNA codes for 1 amino acid, and that's called a codon. So 3 base codons in DNA are transcribed into complementary 3 base codons in the RNA. Then the RNA codons are translated into amino acids to form a polypeptide. So it's a 3-letter code. The genetic code is 3 letters. 3 letters in the gene codes for 3 letters in the RNA will code for 1 amino acid in the polypeptide. And there it is. There's the DNA strand. Here's a triplet code AAA, and it will be a UUU in RNA, remember, because there's no T in RNA. And that UUU will code for 1 amino acid. Then the next one is CCG. That will code as GGC in RNA, which will code for 1 amino acid, and so on and so forth. It's 3-letter code. So there's 64 3-letter codes. If you have four different letters put in triplet codes, there's 64 different combinations. So it's 64. You only need 20 though. So it's like 3 times as many. So this is called a redundant code, because we're going to find out that for most amino acids, they are coded for by more than one codon. Will code for the same amino acid, because you have 3 times as many. That's what's called a redundant code. So genetic code is a set of rules that converts a nucleotide sequence in RNA to an amino acid sequence. The 24 triplet codes, 64, like I said, there are 64, 4 to the 3rd power. 61 of them code for amino acids, and 3 are stop codons, instructing ribosomes to end the polypeptide. Remember, I said you have that punctuation. So the 3 stop codons are the period at the end of the sentence. Tell the ribosome, this is where you stop. This is the end of the message. What about the capital letter? Well, there's one particular codon that does double duty. It's AUG. That's not only codes for amino acid called methionine, it's also the start codon. So it's the capital letter in the sentence as well. So it does double duty. So a given RNA triplet always specifies a given amino acid. So I already answered this checkpoint question. How many nucleotides are necessary to code for a polypeptide that's 100 amino acids long? Well, you would initially think, well, 300, at least 300, because every amino acid needs a 3-letter triplet code. So it's 300, but it's actually 303 because you need a stop codon. So that's a little tricky. You need 100 to code for every amino acid in the protein, and then you need 3 more, a stop codon, to tell the ribosome, okay, that's the end of the message. And there is the genetic code. Now, there's also a square version that I like more than the circular version, but this is how you read this thing. Pick a triple code. Let's say AGU. Okay, what does AGU code for? You start here. That's the first letter, A. Okay, then you go to the next ring, G. So it's AG. And then you go to the third one, U, serine. That's what it codes for. That's how you read this thing. And notice that AGU codes for serine, so does AGC. So remember, it's a redundant code. Most amino acids actually are coded for by more than one codon. There's one exception. I think there's only one exception, and that is, notice, there's 2 exceptions. Tryptophan over here, I think, is only coded for by UGG is tryptophan. And over here, methionine, AUG is methionine. And that's also the start codon. Now, it's a little modified when it's a start codon that tells the ribosome, here's where you start the message. Because sometimes you can find methionine within, it's just an amino acid within the protein. And there it's not the start codon. So it's a little modified. I think there's more methyl groups put on it or something to show that this is the start codon. It's not just a regular methionine, it's the start codon. And then here are the periods, stop codons, UAA, UAG, and UGA are stop codons. They don't code for amino acids. They tell the ribosome, this is where you end the message. It's the end of the message. Protein is finished, ribosome comes apart, and the protein drifts away and does its job. Okay. There is the rectangular methods, which I think is a little easier to use. The first base is, let's do that thing, same thing, AGU. Where's AGU? First base over here. So there's A, so it's in this row. G, it's got to be in this column. So it's in this square somewhere. And there's U, Siri. See? Now, if I can go over here, how about ACC? A here, C here. So, so ACC is 3NA. See, and look it up here, serine. Actually, serine has 6, I think it's the most. Some amino acids are coded for by 6 different codons, some by 2, some by 4, and like I said, a couple of them just by 1. I think it's tryptophan and methionine only coded for by 1. So first, second, third base. Okay. Like baseball, first, second, and third base. Okay, the genetic code is nearly universal. We could say it's universal. Shared by organisms from the simplest bacteria to complex plants and animals. Everybody uses the same thing. So it's really old. It was developed sometime, I don't know, probably a couple billion years ago, and now we're sort of stuck with it. Well, that's the next sentence. The universality says it must have arose very early in evolution, was passed on to eons to all organisms who live on Earth today. So since they share a common genetic code, it's possible to program one species to produce a protein from another species by transplanting DNA. That's how we can make all these human proteins like insulin for diabetics, like clotting factors for hemophiliacs. We put that gene into a bacterium. The bacterium expresses that human protein, and we collect it from there. So it's revolutionized, you know, and really brought the cost down and made it really a lot more supply of all these important human proteins that are being made by bacteria. So many useful genetic engineering applications in agriculture, medicine, and research in order to make transgenic organisms. So there's a mouse there expressing a foreign gene, a gene for fluorescence that was gotten probably from some algae, and they took it from some algae and they injected it into a mouse embryo, and now the mouse expresses this algae gene and makes fluorescent legs. So it's a brave new world here of transgenic organisms, and there's a lot of good coming about from it, but there might be a lot of bad things that might show up. So it's a big grand experiment right now. I hope not too many bad things happen. Okay, transcription is a transfer of genetic information from DNA to RNA. As with DNA replication, 2 DNA strands must be first separated at the place where the process will start. In transcription, however, only one of the DNA strands is used as a template for the other RNA molecule. The other strand's unused. It just exists to hold things together in DNA. So we unwind DNA and we only copy one strand. It's called the coding strand. We copy that one strand and make it into an RNA molecule. The nucleotides that make up the new RNA molecule take their place one at a time along a DNA template by forming hydrogen bonds with the nucleotide bases there. So the RNA nucleotides follow the usual base pairing rules, except that U rather than A pairs U rather than T pairs with A. And the RNA nucleotides are linked by a transcription enzyme called RNA polymerase. So DNA polymerase copies DNA to DNA. RNA polymerase makes an RNA copy from DNA. And remember, these are not nucleic acids. RNA polymerase is a protein. It's an enzyme, so is DNA polymerase. So the start transcribing signal is a nucleotide sequence called a promoter, which is located in the DNA at the beginning of the gene and specific place where the RNA polymerase attaches. So the RNA polymerase recognizes the promoter and attaches there. And this is called initiation. And it's the start of RNA synthesis. During the second phase of transcription, called elongation, The RNA goes longer and the RNA strand peels away from the DNA template, allowing the two separated DNA strands to come back together. So you open it up, you transcribe the one strand of the DNA, you put it back together, you put it back together, DNA back together, and you put the book back on the shelf. Okay, during the third phase of transcription called termination, RNA polymerase, which is a special sequence called the terminator. So it's a promoter starts the process or tells the RNA polymerase where it starts. The terminator tells the RNA polymerase it's the end of the message. You stop there. And then the polymerase will detach from the RNA and the DNA strands rejoin, and then we put the book back on the shelf. So how does an RNA polymerase know where to start transcribing a gene? It recognizes the promoter, all right? So here's transcription. RNA polymerase comes along, DNA initiates it, elongates it, finds the terminator, detaches, and there's the completed RNA. So that's transcription. So nearly every organism on Earth shares the identical genetic code, indicating that this scheme arose very early in the history of life. What major theme illustrated by this? Evolution. So this thing evolved and it's been passed on through billions of years to us now, and we're all using the same scheme. Okay, this is kind of contradictory. In the cells of prokaryotes, the RNA transcribed forms a gene immediately functions as messenger RNA. The molecule translated into protein. No, that's not right, because it contradicts itself, and in the very next bullet contradicts that. You have to modify the RNA that's first produced. I'm calling a primary transcript, and most people call it a primary transcript. The first RNA molecule that's made by transcribing DNA. So this transcription happens in the nucleus and processes the RNA transcript. So remember that. So it's called a RNA transcript or primary transcript. And it has to be processed before it becomes a mature mRNA. So it contradicts itself right here. But so one kind of RNA, so this is called RNA processing, turning a primary transcript into a mature mRNA molecule. One kind of RNA processing is addition of extra nucleotides to the end of the RNA transcript. It's called the cap and the tail. Marks the RNA for export from the nucleus and helps the ribosomes recognize the RNA as mRNA. All right, so we have a cap and tail. Another type of RNA process can be necessary in eukaryotes by non-coding stretches of nucleotides that interrupt the nucleotides that code for amino acids. It was a bizarre when it was first discovered that most genes of plants and animals include such internal non-coding regions called introns. They're nonsense sequences. They don't code for anything. They're called introns. The coding regions, parts of the gene that are expressed, are called exons. Exons with one X. So it's like you were reading a sentence along and it made sense, and all of a sudden it went to gobbledygook. It had no word. It's just meaningless letters. And all of a sudden down here, finally, it picks up again where it left off over here. Now it starts making sense and it links up to over here. And so eukaryotic genes are discontinuous. They're mixtures of nonsense regions called introns and coding regions, sense regions called exons. So when the RNA polymerase transcribes both the exons and the introns, so the primary transcript is a mixture of meaningful sequences and gobbledygook. So these introns have to be removed to produce an mRNA molecule with a continuously coding sequence. And this cutting and pasting process is called RNA splicing. Believe the play has significant role in humans, allowing approximately, well, this says 21,000. I thought there was 30-something thousand genes in a human being. But we can make billions of different proteins. how can we make billions of different proteins if we only have like 30-something thousand genes? Well, that's because you can mix and match exons. You're gonna splice the primary transcript, and you're going to use certain exons and splice those together. If you splice these particular exons together, you make this gene. If you splice a different number of exons together, you make a different protein. So each gene can actually code for several different proteins. I would like to compare it to poker. So you have 52 deck of cards. Think of the 52 cards as 52 different exons. And you're making a protein that's 5 exons long, a poker hand. So you select 5 cards out of the 52 for your poker hand. How many different poker hands can you make from 52 cards? I'm not sure how many, but it's a lot, all right? So here's how you can mix and match exons to make different proteins, to make billions of proteins when you only have about 30-something thousand genes. Okay. And there it is. So DNA, the dark blue is exon, the light blue is introns. So we add the cap and the tail first, then we have to remove the introns and take certain exons and splice them together. And then you have a mature mRNA. And here's the thing that's not meant, so this primary transcript is not mentioned in the book, so you should remember that. First you have a primary transcript, and then it has to be RNA processing. Cap and tail have to be added, and the introns have to be spliced out, GNA splicing. And here's the thing that's not mentioned in the book. It's a very interesting enzyme that does the splicing, that cuts out the introns. It's called a ribozyme, R-I-B-O-Z-Y-M-E, ribozyme. It's a very interesting enzyme, a very ancient enzyme. It's interesting because it's an RNA molecule. It's not a protein. Before, we talked about, in the enzyme lab, we talked about proteins. All enzymes are proteins. Almost all. There's some left over. This is like the one RNA enzyme that's left over. It's thought that when life first evolved on Earth, this whole thing about genetic code and having things move from nucleic acids to proteins, it's all really complicated. You would think that this would just arise out of nowhere, right? Or arise at once. So it was thought that in the very earliest life, proteins actually didn't exist. It was just nucleic acids. DNA for the storage of information, and RNA to do the enzymatic roles. Remember, enzymes are necessary for life. So it's thought to be an RNA world back then, that RNA did the enzymatic rules. But then somehow proteins were invented. It seemed that the proteins did a better job of being enzymes, doing enzymatic rules, than RNA did. So they supplanted the RNAs and took over all of the enzymatic rules, except for the RNA splicing. This ribozyme did it the best, couldn't be replaced, and it's still with us. So it's probably a really ancient enzyme. Okay, so then we're going to go into translation. It's a conversion between different languages, from the nucleic acid language to the protein language. It involves more elaborate machinery in transcription. Remember, so we have to go from a language that has 4 letters to a language that has 20 letters. So the first important ingredient required for translation is the mRNA produced by transcription. Once the mRNA is present, the machinery used to translate mRNA requires enzymes and sources of chemical energy other than triphosphate. ATP, takes a lot of ATP to make a big protein from a bunch of amino acids. And translation requires ribosomes and two other forms of RNA, one called ribosomal RNA that's found in ribosomes, and one called transfer RNA that brings amino acids to the ribosome. So we're going to end it here. So this will be a 106 DNA 1, and then we'll pick it up in the next video lecture for this extra credit chapter and talk about translation. Okay, and also mutations and a little bit about viruses. So it may take at least one and may take two more lectures for this chapter. Okay, so I'll stop now and I'll catch you in the next video lecture here.